Hi everyone, I'm Shiv Kumar, Assistant Professor, Department of Pharmaceutics, Sri Shanmugha College of Pharmacy. Today we are going to discuss about technology transfer. First, introduction. What is technology transfer? It is a logical procedure to transfer a manufacturing process, its documentation, and professional expertise from one site to another. For example, from the development lab to the factory. For a successful transfer, we need a planned approach, trained staff, a quality system, and a full documentation. Usually, there is a sending unit that gives the technology and a receiving unit that gets it. Next, terminology. First, API. API means active pharmaceutical ingredient. This is the actual drug substance that provides the pharmacological activity to treat or prevent a disease. Second one, good manufacturing practices means GMP. This is the part of quality assurance that ensures products are consistently made to the required quality standards. Next, we have process validation. The process validation, which is documented proof that a process will consistently produce a quality product. And next, quality assurance, QA. Quality assurance covers everything that influences product quality. Next one, QC, quality control. The quality control involves the actual testing and the sampling to ensure materials meet their specifications. Next one, quality risk management. The quality risk management is a systematic way to assess and control risks the product's life. As mentioned before, the sending unit is where the process comes from and the receiving unit is where it's going. We'll also use SOPs. SOPs means standard operating procedure. The standard operating procedures, which are written instructions for general operations like cleaning and equipment use. Next one, technology transfer report. A technology transfer report summarizes the entire project, including results and any deviations. Next one, validation. Validation is simply the action of proving that the method actually leads to the expected results every time. Next one, technology transfer protocol. Yeah, the transfer protocol is the roadmap. It must include objective, scope, key personnel, and their responsibilities, a parallel comparison of materials, methods, and equipment, the transfer stages with documented evidence that each critical stage has been satisfactorily accomplished before the next commences, and identification of critical control points, experimental design, and acceptance criteria for analytical methods, information on trial product on batches, qualifying batches, and process validation, change control for any process deviations encountered, assessment of end product, arrangements for keeping retention samples of active ingredients, intermediates, and finished products, and information on reference substance where applicable. And conclusion including signed off approval by the project manager. Next, when moving from R&D to production, the receiving unit must have the capacity to handle the volume. The sending unit must share technical expertise and identify any issues, meaning potential problems in the process. So the receiving unit can prepare. The three stages are there. One is processing, another one is packaging, another one is cleaning. The stage processing. Yes, starting materials. The specifications and the relevant functional characteristics of the starting materials, APIs and excipients to be used at the receiving unit should be consistent with materials used at the sending unit. Any properties which are likely to influence the process or product should be identified and characterized. Active pharmaceutical ingredients. The sending unit shall provide the receiving unit with the open part of API master profile or drug master profile or equivalent data. Information data should follow quality risk management principles. Manufacturer details and the supply chain, step or stages of API to be transferred, synthesis flow chart, physical form of API, solubility profile and pH, partition coefficient and intrinsic dissolution rate, particle size and distribution, bulk properties, water content, hygroscopicity, microbiological quality, and quality and stability requirements. The release and the end of life specifications, stability study summary and a retest period, synthetic impurities and degradation products, potency factor and input quantity justification, storage and handling and sensitivity. Next, excipients. Excipients are just as important because they affect the final product's performance. We must transfer their specifications, functional roles like whether they are a preservative and physical properties like viscosity or microbiological quality. Particle size distribution, dissolution behavior and compaction properties, density, porosity, melting point and pH range, viscosity, osmolarity and moisture or water content, microbiological quality, and finally release and end of life specifications. Information on process and finished pharmaceutical product. The sending unit shall provide detailed product characterization to the receiving unit. This includes health, safety, and environmental information, and manufacturing process and in process controls, control methods and specifications, packaging components and configurations, safety and handling considerations. Next, process development history, clinical development information, scale-up activities, process optimization and critical process parameters, critical quality attributes, pilot scale development data and batch history. The sending unit shall provide information on health, safety and environmental issues related to manufacturing process. The receiving unit shall identify differences in facility systems capabilities, communicate differences to sending unit and assess impact on product quality based on sending unit inputs. Receiving unit should assess its manufacturing and packaging capability, development plant operating procedures and documentation, ensure readiness before start of commercial production. Next on packaging. Packaging transfer follows the same logic. The sending unit provides specs for containers, labels, and anti-counterfeiting measures. The receiving unit then performs studies to ensure the packaging protects the drug and is compatible with it. The packaging information to be transferred, specification of container closure system, design, packing, processing, and labeling requirements, tamper evident and anti-counterfeiting measures, specification for drawings, artwork, and packaging materials for QC testing. The receiving unit shall perform suitability studies for initial qualification of packaging components. Packaging is suitable if it ensures adequate protection against environmental degradation, safety, compatibility with pharmaceutical product performance.